this lecture we are going to talk about Hymenolepis nana and Diphyllobothrium latum, both cystoids. First, Hymenolepis nana, geographical distribution, worldwide parasite, is a cosmopolitan species, more common in warm and temperate zones. Larvae also inhabit the small intestine, so there is no intermediate host. Morphology: this is the smallest tapeworm in man, 1 cm long and 1 mm wide. All segments are broader than long. Each mature segment has 3 globular testes arranged in a horizontal plane and a bilobed ovary in the center. The gravid segment appears followed by sac-like transverse uterus. The scolex has 4 cup-shaped suckers, 1, 2, 3, 4. Also, retractile rostella are armed with a single row of hooks which have a long handle. Life cycle: there is no intermediate host. The mature worm will pass gravid segment that rupture in the small intestine, liberating the eggs. An egg is 50 micrometer. A void contains anchosphere surrounded by transparent embryophores that has 2 polar thicknings from which arise 4 to 8 slender filaments, as you see here. These filaments. The embryo is mature and has 6 hooks. 3 pairs hooklets. Man get infected by injection of the ova that rupture, releasing an embryo that penetrates the villi to live there for 4 days. Change to cysticercoid larvae that rupture the villus to enter the intestine to be fully mature worm within 10 to 12 days. As you see in the life cycle, actually this egg as diagnostic and effective stage at the same time. Either taken by the man as embryonated egg ingested by humans from contaminated food, water, or hands, then in the intestine of the man, the egg, the embryo or anchosphere hatches, cysticercoid develop intestinal villus, develop into scolex or adult worm. Then the adult is in ideal portion of small intestine or ileal portion of the small intestine. This can be released throughout the genital atrium of the gravity proglottid. Gravity proglottid can also disintegrate, releasing eggs that are passed in stools, as here. Also, autoinfection can occur if eggs remain in the intestine. The eggs then release the hexicant embryo, which penetrates the intestinal villus, continuing the life cycle or the cycle. Here, sometimes the egg ingested by insect, then cysticercoid develop in insect. A human and rodent are infected when they ingest cysticercoid infected arthropods. This is the life cycle of Hymenolepis or Hymenolepis nana. Symptoms: a few worms may cause no symptoms. Big numbers will cause the children nervousness, abdominal pain, diarrhea, insomnia, and even convulsions. Eosinophilia as common, finding 8 to 16%. Diagnosis: presence of characteristic eggs in the stool. The fifth example of parasitic cystoid, Diphyllobothrium latum, or fish tapeworm or broad tapeworm. Diseases: Diphyllobothriasis or fish worm infection. Geographical distribution: it is common around areas of Great Lakes, also in Europe, Asia, and Canada. Habitat: the adult stage present in small intestine of man, cat, fox, and other Mammals. Morphology. The adult form length or adult morphology length about 3 to 10 meters, width 2 centimeters, composed of many segments, may reach to 4,000 segments or proglottids. Head or scolex contains 2 potria. No suckers. Note segment length is less than its width. The mature segment is much wider than its tall and contains roster-shaped uterus, as you see here. The uterus is present at the middle of the segment. There is a uterine floor through which eggs are discharged. The female genital organ is formed of bilobed ovary. The testis numerous, occupy the dorsal part. The egg has a operculum, as you see here, and polar knob on opposite ends of the egg. As you see, this is mature segment. Mode of infection. Man will eat undercooked or raw fishes in the small intestine. The blerocercoid larva will stick to the wall to fully mature within 5 weeks. That start to pass eggs and even segments. Life cycle. The eggs are laid immature to be developed in water. It is operculated unlike other cystod, golden yellow. It needs 18 days, then it hatches, releasing the ciliate stage called coricidum. Within 12 hours, the coricidia are ingested by the first intermediate host, which is a cyclop, develop into a procercoid larvae in the body cavity of the crustacean. They need 2 months to be infective for the second intermediate host. Small fish will ingest an infected cyclops. The larva will penetrate fish gut to settle in fish muscles to liver tissues, develop into plerocercoid larvae. So the small fishes are the secondary intermediate or second intermediate hosts, while these small second intermediate hosts can be eaten by a large predator species fish that then serve as paratenic or transport hosts. Large fish like salmon will ingest a small infected fish, and the plerocercoids will migrate to salmon mussels, stay there till man or animal ingest the infected mussels. The larva will develop in the human or animal intestine till migrate and start oviposition. As you see here in illustrated life cycle. Here, when unembryonated eggs pass in feces of the human in the water, eggs embryonate, and then the coricidia hatch from eggs and ingested by crustaceans, which is cyclop, procercoids. Then the coricidium transform into procercoid larvae in the body cavity of the crustaceans. Infected crustacean ingested by small fish, water fish. Procercoid larvae released from the crustacean develops into plerocercoid larvae. Then these small fishes taken by the predator fish like salmon. When the predator fish eats infected small fish, the human ingests raw or undercooked infected fish. Inside the intestine of the man, they change into the adult tapeworm. Pathology may be of no symptoms, but symptoms like hunger pain, dyspepsia, colic, diarrhea, secretion of toxic materials, anemia due to that parasite absorbs a huge quantity of vitamin B12. These symptoms are common among the infected people. Diagnosis: discovery of the eggs in patient stool, or discovery of a segment has a central brown uterus and absence of marginal pores. Prevention: fish should be thoroughly cooked. Good healthy sewage disposal system in endemic area. Human sparagnosis. Sparagnosis is a parasitic infection caused by the Diphyllobothrium or plerocercoids larvae. of the genus Spirometra, including Spirometra mansoni, Spirometra ranarum, Spirometra mansonoides, and Spirometra erinaceus. The infection is transmitted by injection of contaminated water, injection of a second intermediate host such as a frog or a snake, or contact between a second intermediate host and an open wound or mucous membrane. Humans are accidental hosts in the life cycle, while dogs, cats, and other mammals are definitive hosts. Capable freshwater crustaceans are the first intermediate host, and various amphibians or reptiles are second intermediate hosts. Once a human becomes infected, the pleurocercoid larvae migrate to subcutaneous location where they typically develop into a painful nodule. Migration to brain results in cerebral prognosis, while migration to the eyes results in ocular prognosis. We noticed that Diphyllobothrium latum, that fishes harbor the larval stage with tissue damage, and man harbor the adult form with less damage. If man harbor in his muscle the larval stage, this is called the human prognosis. In Far East, a parasite is called Diphyllobothrium mansoni. The larval stage may reach the human body through many methods: application of infected frog or human skin as poultice on the eye, skin, or vagina; injection of infected cyclope where the larvae will penetrate his intestine to the muscle or subcutaneous tissue. This is the life cycle of prognosis, as mentioned before, the same as Diphyllobothrium latum. But prognosis means the development of pleurocercoid larvae in the animal, result in many damages, as mentioned before. As here, embryonated eggs pass in the feces, eggs embryonated in water, Chorocidea hatch from the eggs are ingested by crustaceans, pleurocercoid larvae, the body cavity of crustacean, infected crustacean ingested by second intermediate host, fish, reptiles, amphibians. Pleurocercoid larvae raised from the crustacean develop into the pleurocercoid larvae, as you see here. Predator eats infected second intermediate host, either fishes or frogs for the animals, cats, and these are the stages of the development of pleurocercoids in the animals and even the human. Pathogenesis: the larva will invade the subcutaneous tissue, producing marked inflammations. When the lesion ulcerated, it reveals shiny swelling showing the larva. When the larva die, pus-like substance will be formed. These are the symptoms pathogenesis of the development of larvae in the animals and the human. Diagnosis: in endemic area, that is in China, opening the lesion and finding the larvae.